Welcome to Making Multimedia Reality in your course. Today we have Tasha Biesinger from the Department of Microbiology, Tim Jensen from the School of Writing, Film, and Literature, and Phil Pepe from the Department of Integrative Biology.
Awesome. Cool. Welcome, everybody. I'm going to get started right here. So I need to give you guys a little bit of background on my course for my need for multimedia in my course. So I teach MB 230. It's an Introduction to Microbiology course. There are no prerequisites for this course, and the eCampus version of MB 230 serves a lot of non-traditional students who are currently in careers, often in the nursing field, and they need more certification, and part of that certification requires that they take a microbiology course. So many times, oftentimes, they haven't been in school for very long, or for a long period of time. They're coming back, taking 1 or 2 courses so they can move forward in that certification. Because it's required for these professional programs, the lab component of this course is still on campus. They are required to take on-campus labs, hands-on labs. So because it's an eCampus course and it's taught in the summer during a 4-week period, but also during winter term, during a 10-week, we needed to figure out a way to get these students here to campus with minimal distraction to their careers and their jobs and their lives that they're currently at. Because this is an eCampus course, they really are expecting to take this online, but because the hands-on labs are required for their programs, they have to come to Corvallis. So what we did was we took our normal term, 10-week term where they have 2-hour labs each week, and we crushed them down into 2 days from 9 to 4, okay? So these people come to Corvallis for 2 days, and we try to get these 2 days as close as possible so that they're only traveling once. Some people are traveling from far away. And we've crushed all of the material into 2 days. Okay? That's a lot of information, a lot of material. There's a lot of techniques they need to learn. And normally over 10 weeks, we can teach them this one little technique, and they can make it better, and then they can learn this technique. They get 1 day to learn all of the techniques, implement all of those techniques, and then the second day they look at all of their results and interpret and all of that stuff. So it's very intense, these labs. Okay? So how did we go about helping these students learn these techniques in these 2 lab sessions? And the answer is actually multimedia. What can we help them learn and visualize before coming to the actual lab so that the labs are not quite as crazy and intense? So what we did is we partnered with eCampus and figured out what our core skills were that they really needed to master, and then we developed these multimedia things, which the core skills include aseptic technique, how do you get bacteria, only this one bacterium when there's bacteria everywhere, how do you transfer that from one place to another, the Gram stain, which is core microbiology stuff, everybody loves the Gram stain, streak plating, spread plating, and then we wanted to walk them through visually through eCampus, through this multimedia, one full exercise, so that when they came to lab, they already had an idea in their head, a picture in their mind of what they were going to be doing. Okay. So to this end, working with eCampus, we did develop some videos, but more importantly, some interactives. And we provide these as preparatory tools. They have them on eCampus. There is no grading. It really is just a service for these students. And you would think, well, why in the world would they even look at these things? I'm amazed that they do, and I'll show you some comments a little bit later on. But, okay, so how did this work? Because whenever I come to these meetings, I'm like, yeah, that sounds all nice and good, but how much work is it really going to be? I'm not a multimedia person, so how much do I need to know? So I want some online activities for my students to work through stuff like this. And you can't see it very well. This is what I went to eCampus with. It's a hand-drawn image on a piece of paper, and I am not an artist, I'm in science. So I've got some tubes, I've got an inoculation loop, and a Bunsen burner. And I went to eCampus with about 5 pages, just like this, and said, I want multimedia that shows this. I really, really did. This is what I did. I'm walking you through it, okay? I want videos of this, I want interactives of this. I don't know how to do it, but this is what I want, okay? So I filled out an eCampus proposal thing because, sometimes they give you money for doing this stuff, which is really cool, to upgrade my course. I wanted to develop some multimedia, so I put in a, proposal for some help and got it funded and began working with eCampus. Gave them my papers, and there's my papers again, refresh. I sent some emails to them, they sent me some emails, had a few meetings just so they could say, we're working on this is what we're doing, is this still on track with what you had in your mind? And I said, yes it is, or not quite, tweak this a little bit. But I just had a few emails, few meetings, they took it all. I had a videographer come to lab once, which was great because it was hard to find that one time for him to just come. And ta-da, got some stuff back. So on my end, it actually was very simple. I had to have the idea of what I wanted and had to be able to explain that to eCampus so that they also had that picture, but then they did all of the work. They sent it to me for reviewing. I got to say, oh, I don't like that, or that's incorrect, or change that. And I developed these things. So if I can get out presentation mode, I will show you a couple of them. So this right here is lab exercise number 6. So I told you we wanted to be able to walk the students through an entire exercise. This particular exercise is all about using aseptic technique to get one bacterium from one place to another. And what order do you do it in? When do you flame things? And so we give them the instructions. This is all the instructions they get. And then they click to start the exercise. And they have a lab manual, so they can be reading in their lab manual. It says to do this. How do I do this? Well, let's see. I need to turn on the Bunsen burner first, maybe. Okay. I need to pick up my loop and get a loop full of bacteria. So my bacteria are over here, I have my sugar tubes over there, and of course none of this makes any sense to you, but if you've got the lab manual right there, it says take bacterium number 1 and put it in tube number 3. So they pick up a loop full of bacteria, but I didn't actually sterilize my loop. And so if I do this process, it's going to give me errors. So that way they can really just walk through the lab manual, walk through this exercise, and say, oh, okay, that's right, I missed that step. I need to sterilize my loop. Okay? And they can repeat this as many times as they want. And they don't need to get it right. This isn't graded. It really is just to give them a sense of what they need to pay attention to. What are the key parts of the experiment that they can mess up on? And if they mess up on it, it'll give them a little answer in here. Say, oh, you did something wrong, try again. So they just go through this as many times as they need to so that they can get a good sense of when I'm in the laboratory and I'm picturing in my head, I have my loop and my bacterium, right, and then I need to put it in this tube. So it really is just to give them that sense of what they're doing. One of the critical aspects of this lab is working with 2 hands. And this is my favorite interactive that eCampus developed for me. So again, little instructions. We need to make a streak plate. And making streak plates, there's a lot of flaming of loops to sterilize things. So how do you do all of this? It involves 2 hands. So in one hand, you're going to have an inoculation loop. In your other hand, you're going to be picking up tubes. So this particular interactive has the right hand and a left hand. So we've got a loop in our right hand. We need to pick up the tube in our left hand. We need to take the cap off. Where do you put the cap? Because bacteria are everywhere, so they have many options here. They can set the cap down here. They can set it down on the desktop. But really, they shouldn't set it down anywhere. So they can go through all of these different scenarios just to get a sense of what they need to be cognizant of. There's microorganisms everywhere. They are on your hands, they are on the bench tops, they are on everything. Okay? So how do you work with holding a lid in one hand, a tube in your other fingers of that one hand, and a loop over here, and you need to flame things. So flame your loop, get it nice and red hot. They actually need to hold it in the flame for a certain period of time before it turns red, because that's how long you need to hold it there. And then they need to flame their tube cap, so that also gets red hot. And then, there we go. And then they get a loop full of bacteria. So now they have bacteria on their loop, and now they need to replace the cap. And so they can really walk through all of these little tiny details that people don't always think about. Okay, and then you can move on to your plate. And I'm not going to show you the rest of this, but there's a lot going on in a microbiology lab when you need to avoid all of these microorganisms. And just reading through a protocol, take the lid off of the tube, get some bacteria out, it doesn't tell you or give you a sense of what all these things are. And so these interactives were developed specifically for this purpose. And let's see if I can, there we go. So I came in with my little sheets of paper, said this is what I want, and this is what eCampus developed for me. It really was that simple. I am not a multimedia person. I'm pretty good at PowerPoint, but I cannot make videos. I cannot make interactives. And just for some feedback from the students, I asked the question, did you find the lab videos and interactive lab links useful in preparing you for the labs? Keep in mind, these were not required. I just posted them on my eCampus course. I said, hey, there's some links if you want to prepare for a lab, and whether they did it or not was up to them. But as you go through these comments, almost unanimously, they're like, absolutely, yes, I loved these things. It helped me prepare so well for a lab. There were a few comments, well, you know, I've had other labs before, so they weren't really all that helpful, but if I hadn't had a lab before, they would have been great. So how to develop multimedia, you don't develop it. You develop the idea, and then you hand it off to eCampus, and then they interact and talk with you and make sure that it's right. So here's my presentation. Yes, they were awesome. Okay. Ta-da.
We can spot the Mac people right away, can't you? All right, well, we'll jump over now to the humanities side of things. In fall of 2013, I was hired as Director of Writing for Oregon State. One of the main duties is running Writing 121, a required writing course for every student at Oregon State. Whether they take it here or not, they have to receive credit for it. It's one of the largest enrolling courses at Oregon State. We run about 45 sections a quarter. That's some 3,500 students that pass through, so you can imagine the course load is pretty big. It's a standardized course. So even though we have some 40 instructors, we need to have the assignment sequence, we need to have the grading guides, everything standardized across. So as we started to overhaul the course, we knew that we also wanted to do eCampus at the same time, so that we had our online courses and our on-campus versions merging and kind of unleashing at the same time, not even unrolling. So one of the things we did in the pilot sequences, we looked at our assignment sequences, and knowing that we wanted to introduce some type of multimedia into a course, we wanted to find the spaces where it would provide the most impact. So we looked for pressure points. Where were students having the most trouble in the curriculum? Where do we see either the biggest variance across grades? Where in the feedback do we see students requesting more help? When we ask instructors, where are your students struggling? So we get all this feedback, and we identified a few spots. One of the biggest, you can see the orange there, is the Critical Analysis Project, which is the backbone of the Writing 121 curriculum. And it's composed of 5 different components. They begin with an initial artifact analysis. They do a very close reading of a singular text. They do an annotated bibliography to introduce them to the reading and research process. Then they integrate that research. Then they meet one-on-one with their instructor. And then they actually write the essay after having practiced all these different steps and kind of evolved through them. So you can imagine if I articulated it as boring as I just did to you all, and you were in a writing course at the age of 18 or 19 roughly, that you would be checked out, right? So we knew that explaining the overall trajectory of the Critical Analysis Project was something that we needed to focus on. It comprises the first 6-7 weeks of the course, right? Out of 500 points, 1,000 total, that's a really big deal. So we needed to communicate it clearly. So we worked with eCampus using course development grant funds to develop a video that would communicate this in a very short amount of time, but hopefully in a way that also got students excited about the project. So I'll show you, this is the first few minutes of a video that articulates it.
We're committed to helping you develop your skills and confidence in analytical writing and to cultivating your overall rhetorical awareness. So we've designed the Critical Analysis Project to catalyze growth in both of these areas, giving you entry into writing, research, and critical thinking at a sophisticated level. The project as a whole is composed of 5 assignments: the initial artifact analysis, an annotated bibliography, the initial research integration, a research conference, and finally, the critical analysis essay. And the project takes place over the span of about 6 weeks. You can think of this project as similar to the exploded view diagrams you might encounter in an instruction manual. We're laying out the parts of quality academic writing, exploring them separately, and examining their connections before putting it all together. Writing is a craft, similar to building a table or cooking a meal. And by isolating parts of the process and looking at key components, we can better see how compelling analytical arguments get created. We've designed the project this way to demystify academic writing and to challenge you to think carefully and critically about your writing as both process and product. We begin the project with the initial artifact analysis, where you'll perform a close analytical reading of a cultural artifact of your choosing. After doing some initial analysis, you'll have a better idea of what issues are being raised by your artifact. To explore and better understand those issues, we turn to research, beginning with the annotated bibliography. This assignment focuses on critical reading, information literacy, and research skills. It will also provide you a grounded introduction to OSU's library and how to navigate its resources. Your research and analysis will mature with the next assignment, the initial research integration, where you are asked to further develop the artifact analysis and integrate your claims alongside those of others, situating your analysis within a larger cultural conversation. After these 3 writing assignments, you will meet one-on-one with your instructor to discuss the evolution of your project and collaboratively strategize a plan for the final stage, crafting a compelling critical analysis essay. This final assignment is the culmination of your project, where you're challenged to take the skills practiced up to that point and then push your writing to the next level of sophistication. The Critical Analysis Project as a whole is worth 500 points, or half of the total course points available. Here's how it's broken down.
So we won't make you sit through the next part. But the idea was to help students visualize what is going to be happening over the next several weeks. This is part of a broader strategy within our Writing 121 curriculum to present really core principles and the standard elements of our curriculum in multiple modes. So you saw the assignment sequence wheel. Well, these infographics, not just the assignment sequence, but where they're placed over the course of the term, grading guides, is designed to help students visualize the different types of writing they'll be doing and getting them excited. And we're also trying to communicate that this is a different writing course than ones they may have taken before. So we're both communicating with the actual content of the multimedia projects, but we're also sending a message about the level of expectations that we expect of their writing. So the actual process itself, we came to eCampus with a rough idea of what we want to articulate, and then just a handful of examples of different multimedia projects we like. Most notably, the RSA Animates you may have seen, where they draw on a whiteboard as they go along. Now, if you've ever listened to a lecture and then seen one animated, with cartoons, you know that your memory recall is extraordinarily better when something is visualized. So we showed them a handful of things we liked. We showed them the script. The next thing we see is a very rough draft. And then actually the process was fairly simple. We said, can we tweak this, this, and this? We wanted to communicate this. Can you change this? Maybe highlight that. And that really was it, which is so different from every other multimedia project that I have worked on collaboratively across. So we worked on our end to make sure that the narrative was correct. And then when eCampus came to us, we simply aligned our concepts. So I will say you just a few minutes of the second bit. The notion here is that we didn't want just to explain the process. We also wanted to provide explanation of the core principles behind it, but then also provide tips for students.
At each stage leading up to the final essay, you will be extending, deepening, and refining your analysis and argument. In other words, the sequence is not one of addition, where you take the initial artifact analysis, add some paragraphs of research, and for the final essay, add an introduction and conclusion. Rather, this project is recursive, with each stage calling for revision in the strictest sense of the word, seen anew.
So we won't go over the rest. The key point here is what we wanted to do is provide materials for instructors that help supplement what they're doing in the classroom. So if students watch this coming into class, they have an outline of what will then be applied and worked through in class. The second pressure point that we found, the first was articulating the whole. The next is the very beginning. What we discovered through the pilot sequences is if you don't choose a correct, like a really great artifact for your projects, then the next 6 weeks of your life is going to be very hard. And if we learn this through various student feedback. So what we decided was really concentrate on that initial moment with the artifact. And so what we did, we made a video.
So what exactly is an artifact? Let's break it down. The word artifact comes from the Latin art, meaning by skill or art, and factum, which means something made. Artifacts are things made by people. They are things that can be seen, read, heard, or felt. In this course and in most of the humanities, artifact is synonymous with text, understood as anything that can be read analytically and interpreted. Here, text refers to more than words on a page. We use artifact in conjunction with text to emphasize that in analysis, you're not limited to things that are only comprised of words. For example, an artifact or text can be a photograph, an essay, or a movie scene. Paintings, presidential speeches, and even cereal boxes are also artifacts that can be analyzed to see how they communicate meaning. Music videos, poems, and plastic action figures are artifacts. They're texts that can be read and interpreted. Public service announcements, car commercials, environmental movement posters, restaurant menus, TED Talks, bumper stickers, government policy proposals. All of these are made intentionally by humans, and they communicate meaning about the culture in which they were created, who created them, and they tell us something about the audience for and users of the artifacts. As you can see, there are a lot of options to choose from. Your instructor will set some parameters on what types of artifacts may.
So what we wanted to do there, you can tell that there's this breathless kind of repetition. We really wanted to expand this notion of the type of artifacts available for students. And the best way for us to do that was through this kind of visual representation of just how many options are available to us, because we were trying to counter the notion that students were feeling already stuck within the parameters of the course, that they had to choose something. What we found, though, is it's not just enough to blow open the doors, that we really then had to model a process for the students. And so we took one example and followed it out.
3. It is specific
These
and
are the
concrete.
tips that we gave to
For
students
example,
for selecting a
the
great
concept
artifact.
of liberty isn't a good artifact because, well, it's not even an artifact. You can't point to liberty. However, a magazine advertisement for a Jeep Liberty that shows the SUV tearing through a forested wilderness, encouraging you to leave the civilized world behind and be free, is a specific text, an artifact you can actually point to and analyze. If the concept of liberty interests you, of course, this is an excellent start. The next step is to look for artifacts where either the word or concept is invoked. Whether it's in a car advertisement, a politician's stump speech, a comic stand-up routine, a university commencement address, you get the idea. It doesn't matter where you begin the journey, as long as you end up with a specific text that you're stoked about.
So
So to recap,
I'll very quickly point out that those examples that they gave, those were all by the videographer Warren Blythe. So he came up with the examples. It was eCampus that actually provided a fair amount of the content for that. They followed a script, we went back and forth, and then we chose the right examples. And I emphasize this point to decrease the notion that you have to come up with absolutely everything for your multimedia with eCampus. It's an incredible collaborative relationship, and they actually bring a lot of productive ideas to the table. So I'll show you one last bit that we worked with within the Writing 121 overhaul, and that's the next major assignment. The Exchange is a student-driven publication. It features writing by 121 students that they determine relevant to the OSU community. So the idea is that students are the ones that decide what is most important. It's published as an insert in the Barometer once a quarter. It comes out at the beginning of week 8. And the key here is that students are not only authors of the articles, but then they act as the reviewers. And then finally, after they take those revision notes, they improve the essay, then they submit it, then they act as editors as well, deciding which pieces should move forward. So they're mimicking the entire publication process. Actually, not even mimicking, they're just doing the entire publication process. Well, to get them started, we worked with eCampus to develop an infographic that would ask them to do some audience analysis, because the core of this assignment is that it's an authentic writing condition, that they are writing for a real audience, that they have the real possibility for change. So the question is, who's your audience? And so this infographic was made with statistics culled from various OSU sites. And the arc here is we purposely chose a smattering of different statistics and kind of mashed them up against each other so that students could find different access points. Now, how this is used in the classroom, they are instructed to read it the night before, they come into the classroom, and then it's the point of departure for a conversation where they can ask questions. Well, what did you find interesting about this? What did you find compelling, right? How might your piece that you were thinking about writing relate to these different valences within our community, right? How might somebody respond to this? And these spark pretty good conversations, because take, for instance, that the Greek population is 13.1%. If you were to ask students what percentage do you think the Greek population is, you get these crazy percentages like, oh, it's like 75%, of course, right? And we all know that 42.7% of statistics are made up on the spot. So what we want to do is counter that notion, actually bring in stats and use this as a catalyst for helping them think about it. So that is actually ranked highest. Now, very quickly, how do we see this moving forward? What we would like to do is, right now they're acting as supplements within the Writing 121 curriculum. Next year we'll be moving to integrate them much more. So we'll be asking students to work with the text a little bit out, then bring them into the classroom and already have some digested notion about the different assignments. And then we work with that more in class. So instead of, do you have questions, watch the video, right? Did you watch the video, enjoy it? What kind of questions do you have? Instead, it's worked into a type of activity that really tries to speak back and forth between the content we've developed and the questions the student have. Thank you.
Hello, I'm Phil Pepe. I'm from the Integrative Biology Department, and I'm going into my second year here. I also work pretty closely with eCampus and have had a lot of really good interactions with eCampus. And I have sort of a big task. It involves developing a whole series of online classes. I'm into working on my fourth one now. And also working in the classroom this semester, and it involves a new writing intensive course, which is entitled Critical Thinking and Communication in Life Sciences. So also tasked with helping out with the development of some online labs for a biology series that is being developed by Andrew Bauma in our department. So I've had to look at this sort of with a broad brushstroke, thinking about developing different multimedia objects and integrating them, and then seeing how much they help in portraying a concept, always starting out with the outcomes for the individual courses, but keeping in mind that I'm probably wanting to get more bang for the buck out of any particular project, because I can repurpose them in various different courses. So I have been looking at the development and involved in the development of online lectures, of solo activities that students are interacting with, and then ones that involve students interacting with each other and with myself at the same time. Right now, one of the projects that is in the works is a rather large video project, which is spotlighting the evolutionary biologists in our class. And these videos are being incorporated or integrated into a course that I'm teaching entirely online, which is Intro to Evolution. It's a backcourt course. So instead of just recording my own videos and lecturing to the students, I chose to basically use the brilliance of all of our faculty, because we have 10 different faculty members in the department that are involved in evolutionary biology. And I was basically able to go into their classrooms and their laboratories and have them do a lecture for the particular week by showing what they're doing. And this whole series will be placed into the course, and then we'll have some interactive discussion about each one of them. And this should start up in summer. Another project, this one has been being developed for use in a whole series of courses, and perhaps in courses outside our department as well. And that is the putting together of a virtual microscope that could be operated in the same fashion that a student would operate a microscope in the lab. Let's see if I can show you how this works. I thought it was here somewhere. That's not it. Oh, there it is. Ah, I lost it. Hey, so this is a mock-up of an actual microscope that we have in the laboratory and that's used in various labs in integrative biology. And up here we have controls for it. That the student actually has to go through the whole process from turning on the lamp in order to be able to see a specimen to focusing and also moving the slide around on the stage. I'm having a little trouble with operating it. You want to help me, Mike?
Player's built for specific resolution, so you're not seeing the entire screen. But if you, where do you have it up down here in the Explorer? So we're gonna have to scroll down a little bit to see all the controls. We should have got you the executable.
Yep.
You can change resolutions in that one, but turn it on so at least they can see the specimen. Okay, and then so you basically have to use the scroll bars to go around because
Yeah, and then
you
I'll
don't
have
have
to
the
move up and down.
executable version. Yeah. So,
Okay, this is going to be a little bit difficult because I don't have the version that will execute on the small screen. But I'll just show you one or two little features of it so that you can see how it works. And that is that you click on the particular part of the microscope that you want to manipulate, and it lights up in blue on the actual scope that you're seeing. And then down below, there's a control. I'm mousing around. I'm just going to skip this because I'm having too much trouble with the mouse pad. But essentially, you can focus the microscope, you can move the slide around on the stage, and you're going to see the image moving around. And you can then change powers and look at it at higher power. Okay. That mouse pad is not going to respond to me. Well, and essentially in this field of view, what we're looking at here are plant cells from a lettuce leaf. And the student can focus in on that. And we're now also developing some views of bacteria and fungi and other types of microorganisms that can be used. Now that the shell is put together, we can take a series of photos of any object under that microscope and then place it into this program, and then the student can manipulate that and look at it. Okay. So I just wanted to tell you a little bit about how I go through the process and what I consider in terms of trying to pick out types of multimedia projects that we do. Okay. And one of those is by basically gauging student responses to those objects. But also I look at them in regards to how those can be used as a teaching tool. And typically it involves some writing, discussion, multimedia responses, or data analysis. In fact, in the course that I'm currently developing, which is a writing intensive course, I have sort of a cross between what Tim and Tasha are doing, because they have to write a term paper, and it involves research, but research of a different kind, and that is experimentation, where they have to work with data. And so as I thought through this, the class itself is a lecture class, so there's no lab component to it, so they can't really develop a data set of their own in the classroom. And in addition, they wouldn't have the whole experience of working through an actual scientific project and paper if they don't have a data set. So essentially, I asked students on the first day of class if any of them were working on a project where that was data-driven, where they had their own data set that they could work on. There were 2 out of the 20 students that had that. So what I provided is a series of different types of experiments that are in a virtual setup, so that they could actually go through generating data and then analyzing it and then presenting it in written form. And the way in which I went about that is, since I've had some experience with multimedia, I worked through kind of storyboarding the lab, which then I got help from eCampus in terms of the presentation, both with video, audio, photography, videography. All of those are involved in it. And they have provided tremendous support. Mike Miller has helped tremendously. Nick Harper has also been involved, as well as Victor, and of course the instructional designers like Melanie. So the student outcome that I was looking for here is one in which there would end up being student-student interaction, that they could work on these projects together, and that there would be interaction with me as the instructor, and then also they're interacting with the content itself. So the end result of one of these projects, which is about enumerating soil bacteria and fungi, okay, involves a virtual activity where they can generate hypotheses, they can collect data, and then they can test the hypothesis using the data, and then they can present it all in the form of a term paper. The project actually started as a live project, which we did on campus with the soils folks, where they had 4 compost bins out, and these compost bins involved having a 2x2 experimental design where they enriched 2 out of 4 bins with nitrogen-rich materials, and then 2 out of the 4 bins were turned over or aerated regularly, so that there were 4 different types of compost to test. So when a student goes into this virtual lab, they get to pick which one of these compost bins they're going to sample and study. They are given some instruction in regards to the actual process through video tools, where we go through, this is how you would do it if you were in the lab. They also go through a demo, that is, this is how you do it in the virtual lab. And then they get to perform the experiment. and generate data. Then they get to pool the data amongst themselves and then analyze the whole data set. And they actually go through the process of virtually looking at and working through the experiment. The experiment involves taking a soil sample and doing a serial dilution, which is in these bottles. And they have to pipette and pass fluids from one bottle to the next to dilute down the sample. And then they use Petri plates, and they have to flame an applicator in order to sterilize it, and then spread the known volumes over the Petri plates. And then they let those grow. During the whole course of this experiment, they can go back and look at the actual video, or they can go back to the demo. And if they hit a point where they've done things incorrectly, they're stopped. And after they complete one aspect of it, then they go to the next step. And then ultimately they have a data set by counting colonies from the growth of bacteria on those plates. So this mimics a real lab activity, and in the end they end up with the same kind of data set as if they had run the experiment themselves. And with a randomly generated result that will basically mimic the data set that we actually got by running this in the laboratory. So in the end, they have to do a bit of math because they have to divide the colony count by the dilution factor so they can, in fact, generate the number of bacteria that were found in the original soil sample. Okay, and they work through this, and then they get a number which they then can add to the data set. Okay, and then in the end, they are able to discuss their results, pool their results, and then write their report. And so they're able to do this in a classroom where there is no lab component, but they're also able to do this entirely online. So the project involves setting up a scenario where the writing intensive course can be offered online with a virtual lab component to it. And we have a couple of other labs that are in development in order to give students a variety of choices. And then these same labs can be utilized in other contexts, so they could be repurposed. And they mimic a real experiment that's been going on campus as well. Okay. Thank you.